This is not what the survival analysis is about, this picture, but maybe it's what you at the very beginning thought about. So in fact, survival analysis is about time to event, and the event is usually death or some disease progression or disease relapse. So as you may understand that survival analysis is very, very important in clinical research, especially when we are comparing several groups of patients. They may have different diseases or they may have different treatment of the same disease. And in this case, when we want to compare the efficiency of treatment, we should do the survival analysis. Also,
Margarita, I'm sorry. Will you start listing slides?
Yeah, I'm just at first introduction. Yeah, it's okay. I'll start listing.
Sorry, please. Sorry.
No, it's okay. And also, apart from the comparing different groups of patients and treatment, we would like to use the survival analysis for identification of some important covariates and biomarkers which influence survival, aging, and other problems. So at the very beginning, I would like to introduce what values and what functions are estimated in survival analysis. And for that, we should recall such statistics things as probability density function and cumulative distribution function. So the probability density function is the likelihood that some value x will take a value exactly equal to x. And the cumulative density function is the probability that some value x will take a value less or equal to x. So in the survival analysis, we are talking about the survival probability, about death probability, and about death density, where death density, in fact, is probability density function, while survival probability and death probability are the cumulative density functions. So in survival probability, it is the probability that some value of death, time of death t, will be more than times in our axis, while the death probability is complementary to survival probability, and it's the probability that times of death t will be less or equal to times t in our axis. And the density is just the likelihood that our values of time of death will be equal to the values x or t in our axis of time. So in this slide, you can see all these functions depicted in one picture. And again, we are talking about the death density and the probabilities, which are the death probability, like the proportion of death area under the curve, and the survival probability or proportion of surviving patients to sometimes t. And now let's talk about one more very important thing in survival analysis, which is the mortality risk. As Dmitry just said, mortality risk is very associated with aging. However, let's just understand the logic which is behind this mortality. So let's imagine that we have some mouse which is 1 year old, and then it is 1 year plus 1 day old. What is the probability that it will die within this time interval, within this 1 day, or within some time interval dt? So for that, you can see this formula, which is in fact the conditional probability formula. So it is the probability that time of death t will be within the interval, given that time t is more than some time. So if we want to estimate this probability for some period dt, we can have the limit, and with the formulas of expressions for survival and death probability, we can get this formula. So again, the mortality risk, or it is also called hazard is the probability of death within some little time interval. Well, and now let's talk about the methods in survival analysis. In fact, they can be divided in two great parts, like statistical methods and machine learning methods. So in statistical methods, we can have non-parametric, parametric, or semi-parametric methods. As you see from their naming, the parametric method has some parameters, and there should be some underlying distribution of times of death t. While in non-parametric, this underlying distribution is not known. There is also the semi-parametric method, which we will briefly discuss a second later. And right now, I just want to say that also there is a huge part of machine learning methods, which we'll also discuss today. In fact, we will discuss the most advanced ones. Right now, I would like to say that all this survival analysis, it has some challenges. At the very beginning, we can say that if we are talking about probabilities, about the survival, it's the regression problem. However, there is a twist, as some of our data is censored. What is censoring? Censoring is not observing some values of event and time until the event for some patients. Why does it happen? The first reason is that our study, it is limited in time, and some patients, they may just not observe this event till the end of the study, and therefore we will not know if they are dead or not till this event. Till this time, we'll just know that they didn't observe the death. And also some patients, they might be just withdrawn from the study for just in some periods. So we also do not know for them if they are dead or not. We just know that until some time they were not dead. And this is very challenging part of survival analysis because this data is missed. However, we cannot just get rid of this data, or we cannot impute it with some values. We can only use it in the analysis, but taking into account that till this time, the patient didn't die and hadn't experienced the death. Okay. Also, there are several challenges, as for example, nonlinear relationship between the covariates and the mortality risk, and so on. And also the high dimensionality of data, and in all these situations, the machine learning methods are believed to have better performance than statistical methods. Okay, and right now let's talk about the semi-parametric method. We will not talk today about the parametric and non-parametric because we have not so many time, and semi-parametric is one of the most widely used methods in statistical part of the analysis. And on these Cox proportional hazard semi-parametric methods, many machine learning methods are based. So it's like must-have theme in survival analysis, and we will cover it right now. As I said, it's the semi-parametric method. Why? Because the distribution of time to event is not required. It is more consistent than parametric methods and more precise than non-parametric methods. That's why it is usually used in survival analysis, in the statistical part of survival analysis. So we can see the formula for Cox proportional hazard model. It consists of two parts, of the baseline hazard and of the partial hazard. You can see that the baseline hazard is in fact population level. It has no dependency on the covariates of the patients or on the coefficients of them. It's just population level, but it depends on time. While on the contrary, the partial hazard, it depends on the patients, but do not depend on time. And X is covariate vector, while beta is the coefficient vector. So why Cox proportional hazard model is semi-parametric? Again, because the baseline hazard is not specified, and therefore we don't know the distribution. Why is it proportional hazard model? Again, because the baseline hazard is population level, and therefore the hazard ratio is independent of baseline hazard and independent of time. Here we can see like the picture of what can be called proportional and what cannot be called proportional. On the left part, there's the proportion of the covariate 1 and covariate 2. And this proportion, it doesn't depend on time. So that's what the Cox proportional hazard model is about. Also, as I told, all of the survival analysis, it's type of the regression problem, not classification problem. I encountered in some articles that they called classification problem survival analysis, but it's not what the survival analysis is about. Survival analysis is about time until an event, about the probabilities, but not just about the classification, death or alive. It's not about the survival analysis. So it's the regression problem. However, in Cox proportional hazard model, the baseline hazard is not specified. And therefore, we can't use the standard likelihood method for parameter estimation. And Cox, who invited this method, he thought that, in fact, this baseline hazard is nuisance function, and we do not want to estimate it while we want to estimate the parameters of covariates. And for that, we can use not just the standard likelihood, but the partial likelihood. So let's just talk how this partial likelihood can be derived. So if we're talking about the individual probabilities, we can see that there is the probability for some patient to die within time interval dt. And if we divide it to all the patients, their probabilities to die within this time dt for all the patients which are at risk, this will be the individual probability. Again, it's very important thing. Here you can see that there are patients at risk. This means that all the patients are estimated, both who have experienced the event at this time and who have not. So if, for example, censoring is occurred, then we're not talking about the death for this patient, but we are estimating this patient as patient at risk. Well, the product of all these individual probabilities for all the individuals is in fact the partial likelihood. And yeah, you can see that this risk thing and also this parameter, they are taken into account censoring. Okay, and so by maximizing this partial likelihood, we can get the parameters estimation. But also for just to make it easier, we can estimate not and maximize not the likelihood, but minimize the negative log partial likelihood. So it's just the formula, this formula just transformed to this log likelihood. Well, and by minimizing it, we can estimate the parameters. And now let's talk about the neural network and how they work with survival data, how they can work with paradigm of survival. So again, as I told you, the survival analysis is about regression, but with a twist, with this censoring data. And accounting for this censoring data is like working with a paradigm of survival. And therefore, we cannot just use some machine learning methods, some typical classical machine learning methods. We should use the methods which can account for censoring, can account for the paradigm of survival. But at the very beginning, let's just recall how the neural network works. So it's the model of artificial neuron with the inputs and the weights for each connection of the input with our artificial neuron. Inside the neuron, the summation occurs of the inputs multiplied by weights and their sum with some bias. And on this thing, the nonlinear activation function is applied. Why nonlinear activation function? Because we have several layers of neurons, and to make the nonlinear combination, we need nonlinear activation function. Otherwise, if it would be linear, there will just be no sense in all our network. So we have nonlinear activation, and by that we have some output. During the neural network training, we are getting Y predicted, so the output of our neural network, which is in fact the function of input and the weights. And then we can compare our predicted value with the true values, and by that compute some loss function. So the loss function will be the function of the true values, of the input values, and of weights. And then we, during the backpropagation in our neural network, we want to define how we should change our weights to make our predicted Y value more precise. So for that, in backpropagation, the gradient descent is done, and we should calculate the derivative of the composition of functions, derivative of loss function for each weight, to get the understanding of the gradient, of the slope of our loss functions. So we should understand by this gradient where we can go to increase our loss function and where to decrease. And our goal is to decrease loss function, because in fact loss function is talking us about the error, and we want to decrease the error. So we want to find some minimum of this loss function. And the gradient for us is talking about the slope, where we should go to increase. So we should go in the opposite direction of the gradient to decrease our loss function. And by some tiny steps, which in fact refer to the learning rate, we are going to the minimum of loss function. So each time we are adapting our weights to minimize the loss function. That's the idea behind all the work of neural network. And now let's talk how neural network can work with survival paradigm, and with tensoring. In fact, they can work with it using the Cox loss function. In fact, as I told you, a Cox proportional hazard is the linear model, while survival neural networks can be considered as the non-linear extension for ComputAge. During this idea, we are estimating the partial hazard, and the partial hazard is the output of our neural network. And then we are trying to decrease the Cox proportional hazard loss, which is the function of the weights. So we are trying to search for such weights which will give us the little loss function, the little ComputAge loss function. And this ComputAge loss function, it gives us the opportunity to work with tensoring and to work with survival. As again, here we have the values which are typical for survival, and it can work with tensoring. And usually in our neural network, we have fully connected layers and some dropout layers to prevent overfitting. Well, and right now, let's talk about maybe the most interesting and however complicated part, the integration of multi-omics data in survival analysis. So in fact, multi-omics data is very valuable, and it can give us information about the genes, and not only genes, but some mutations and alterations and epigenetics marks, which are associated with some diseases, with survival, and with the efficiency of therapies, and so on. However, there are several very important challenges in integration of multi-omics data. First of all, because omics data usually, all the processes within cells, they are interconnected. Therefore, lots of information in multi-omics data are complementary. And there are lots of correlations within multi-omics data. Also, multi-omics data has high dimensionality, which is also the very, very huge problem. Moreover, it not only has the high dimensionality, but it has the problem of big P, so lots of covariates, and small n, which are not so many patients. So we have not so many patients, not so many rows, however, lots of columns, lots of covariates. And also, there is a problem of interpretability because of the black box nature of all neural networks. There are several approaches, classical, like statistical approaches for these problems. For example, the regularization of our COGs, and also integrative clustering on joint latent variable. So latent variable is in fact is the variables in the latent space, which can still give us lots of information of our data, however, be low dimensional. And also PCA, principal component analysis, which also can help us to do the dimensionality reduction, and identification of some correlated models so that we can get rid of some correlation and make our data not so high dimensional. But there are also classical, I will call it classical DL approaches, because in fact, if you read articles about multi-omics integration survival analysis, they can be divided into two parts, into the part of very advanced DL approaches and into the part of very interesting, very intriguing and beautiful biological approaches, however, also with deep learning. So the classical DL approaches to decrease dimensionality is autoencoder, variation autoencoder, transfer learning, and meta-learning. And let's just a little bit discuss these methods, not in the context of survival, but like how they work, at least some intuition. So autoencoder. Autoencoder is the model which helps to encode our data from initial space to the latent space, so that we will have the reduction of dimensionality and noise. For example, you can see this picture of our original data, high dimensional, and then it can be transformed to latent space, which still gives us a lot of information about this data. And usually autoencoder is talking us about two parts. The first part is encoder, which helps to transform our data to this latent space. And the second part is decoder, which helps to have our data back in the initial space, however, with some noise reduction. But in the networks of survival, when we want just to have the dimensionality reduction, we're taking only this part of autoencoder. So transforming our initial data to the latent space, and then giving all these features, like latent features, to, for example, CoX-PH based neural networks. The second approach is variational autoencoder. In fact, as you see, it's also autoencoder, but it's talking the difference between the usual autoencoder and variation autoencoder is that it is learning representation of the data with some parameters, with some distribution, like mean, for example, and variance. And also, if autoencoder is talking about some discrete values, the variational autoencoder is talking about the probabilities, and it addresses the issue of non-regularization of a space in a usual autoencoder, and also it helps us to have the opportunity to generate some new like information from the spaces which are which cannot have this generative opportunity in usual autoencoder. Like here in this picture, for example, using usual autoencoder, you can, for example, just reduce some noises in this picture. But variational autoencoder with its probabilities and with its regularization can be used not only to reduce the dimensionality, but also to generate some like new features. And this idea can be used in survival analysis if we want to get some interesting relationships, some new informations from this latent space of our genes and other multiomics data. Then about a bit about transfer learning. It's the thing when we have different tasks and we transfer our some learned information from task 1 to task 2. So just a second. So for example, we have some data in task 1 and we're making some model, and then we can take this model, this pre-trained neural network, just without the last layers, and put here other last layers, and this neural network will work with your other task, with other data. Just because the first part of it, during training on other data, it just like got information about these types of data. It could train better because it just has better ability to extract some features from such types of data than neural network which was not trained on any data. So it has usually better performance than just direct learning on some data. But of course, to apply this transfer learning on multiomics data, you should consider that your data sets should be quite similar, maybe some close, for example, types of cancer and so on, so that the data sets should not be too different, because in such case it may not work. And the meta-learning part, which is highly used in articles and in modern working with multiomics integration. So it's also about different tasks. So at first, let's just see this picture at the right part. For example, we have task 1, and we predict like the labels and can have some loss. And we have some different task 2 where also we have some losses. And by combining these losses, we can have some meta loss. And in meta learning, we would like to minimize meta loss so that adapt all the weights both in this task and in this task, because we have the same model, the same network here and there, and we are trying to use this meta loss on different data sets. So in fact, in case of multiomics data, we can do this meta learning on different parts of omics data sets, like combining these different omics data sets as different tasks. Well, let's talk about one of, I think, a great article where meta-learning was used. It is called Interpretable Meta-Learning of Multi-omics Data for Survival Analysis and Pathway Enrichment. Here they used 17 different cancer types from TCGA databases, and each cancer type was as a task. So as a task here. Like here we have animals and fruits, for example, but there we have different data sets of cancers. And as the multi-omics data, they had transcriptomics, proteomics, and clinical data. Also, as I told you, that many neural networks, they work with paradigm of survival because of the Cox proportional hazard loss, and this neural network also has this Cox proportional hazard loss as the main function to compute the errors and loss. They do the meta-learning with the Cox proportional hazard loss and get the hazards. Also using the algorithm of deep lift, which is in fact backpropagation algorithm, they can get the scores for each features, for each covariates, and use these scores to sort the genes and to apply for them some enrichment and pathway enrichment analysis using, for example, profiler or GCA. About the feature importances, here I said that they used backpropagation. In fact, this search for feature importances can be divided into two parts. Like forward propagation and backward propagation. Forward propagation is the permutation test, which you, I think, heard about, or permutation test is about shuffling some covariate so that you ruins its connection with the output and see how much the predictive power of your model decreases. If it decreases a lot, therefore this covariate was important. If not, therefore not. And in backpropagation, the principle is absolutely different. You are at first assigning some null values for some neurons, for some covariates, and then your neural network is doing at first forward propagation and then backward propagation, computing the losses and computing how much you should change the weights. So you can blame these changes of weights for this neuron, for this zero value of neuron, and therefore understand that, for example, this value should be changed a lot. Therefore, it means that this neuron is very important, this covariate is very important. It's how it works. Well, so in this article, they made several models like direct learning and different meta-learning models, for example, including only transcriptomics, only clinical data, only proteomics, and integrating, for example, all omics data sets. And they computed that the best model was the meta model on all the cancer types, which included all omics data, which is in fact a great result. Yeah, and now, but all these things about autoencoders, about this meta-learning, transfer learning thing, it was just some advanced machine learning approaches. However, there is the biological approach to solve all these problems. And the biological approach is about incorporating some prior biological knowledge and about making models more interpretable. And so it is to what we want to achieve, to make it more interpretable and biological. The first article I would like to discuss is the Salmon survival analysis learning with multi-omics neural networks on breast cancer. Here they used just one cancer type, breast cancer, and transcriptomics, microRNA, seq, and clinical data. Also, their model had Cox-based loss. You can see it here. So it really resembles the loss for the Cox proportional hazard. However, it also has some regularization. And here the minus should be. I don't know why right here you cannot see it, but here's, of course, it's the negative, look like the hood. And what is the biological idea in this article? The biological idea was that they used co-expression and co-expression models, which can be derived by the use of co-expression analysis in some R packages. By that, you obtain matrices. So here in the scheme, you can see that they did the gene co-expression analysis, obtained some matrices, which are in fact the modules of co-expression. And these modules then are going to the network as the inputs. So then the inputs of network is not just the multi-omics data and all genes, all expression, and so on, but the modules of co-expression. And in fact, by doing this co-expression analysis, they decreased the dimensionality. And also, they decreased the dimensionality in the hidden layers. And all these things are going then to the next layers and calculating this Cox loss and the hazard ratio. So that they in this article tried to do the next step by the hazard ratio to divide patients into two groups, like with low risk and with high risk. In this article, they used not the backward propagation as the article before we talked about, but the forward propagation. So they just assigned some modules and covariates. So these were like two different steps how they did it. At first, they assigned modules like the null values and understood which modules were important, which models of co-expression. So it's like the biological knowledge which they get, which models of co-expression were important. And also they assigned some covariates null values. So by this, they understood which covariates were important. Also, they compared their model with some other models. For example, it was greatly better than random forest. And also, they compared it with a Cox and Net model, but don't think that their model was not so good because it has the similar values as Cox and Net. In fact, they didn't use the default Cox and Net. They adapted Cox and Net also with these co-expression modules. So the difference between these two things are just some architectural differences, but all of them use co-expression modules, while random survival forest, for example, did not use these co-expression modules, and it has much lower predictive power. And also they showed that if we include all the multi-omics data, we can have better results of our prediction. And the last thing, the last article of biological approach, which from my point of view is the most beautiful article with incorporation of multi-omics data which I have seen. Also, it is named Deep Omics, Scalable and Interpretable Multi-Omics Deep Learning Framework and Application in Cancer Survival Analysis. In this article, they also use several types of cancer, like the very first one, from TCGA also, and several types of omics data, RNA, methylation, CNA, somatic mutation, and clinical data. It was also Cox-based loss model. You can see here the loss function. It also very much resembles Cox, but it has the regularization of weights. In this article, it a bit resembles the previous article also about biological approach, but they made a huge step in their understanding of biological processes. And they don't just included some modules, but they included some module layer with non-fully connected network. What was the logic? The logic in the previous one, as you can see, was that all the omics data is not interconnected. Yeah, so you can see that the co-expression modules were computed for each type of omics data independently. However, here they thought that these multi-omics data may be interconnected with each other, because methylation, it shows us, for example, inactive gene regions. So, for example, low level of methylation will be where our gene expression is high. And so, for example, by that, there will be interconnected, and so on. And so by that, they included functional module layer in the network. And in fact, those who is doing this approach, those who is running this network, he can himself conclude which omics data should interact and which omics data should be included in one module and which should be included in another module, and so make these connections. And then after this functional module layers, just usual hidden layers and output layer computing hazard are done. Also interesting thing which they did in this article was risk stratification. So as I told you in the last article, they also divided patients into groups like low risk and high risk. But that's all they done. They have no conclusions based on this stratification. But here, just a second. But here they used for this high risk and low risk group the Kolmogorov-Smirnov test with Bonferroni correction to compare these functional module nodes. So they compared each functional module node for these two groups, low risk and high risk, and identified which functional modules had great difference. And by the p-values, they by obtaining scores of p-values, they said which covariates, which features were important and which were not so important. And their method had great predictive performance. So I think that it is very, very beautiful, intriguing biological approach. Well, so the summary is that our multi-omics data is very valuable, but its incorporation is challenging due to high data dimensionality, complementary information, and low number of patients. And moreover, the interpretability of deep learning models is difficult because of the black box nature of neural networks. And so various methods, deep learning methods, are being developed to handle these challenges. But the most beautiful are the biological methods where the prior biological knowledge is incorporated. And the rule is the more biological knowledge you incorporate, the better your model is. So thank you very much for your attention. Join our chat.
Margarita, thank you very much. Dear colleagues, do you have questions?
Thank you very much for your speaking. And one question: is it possible to use the data for different patient groups? No, for example, there are some types of omics data. One group for proteomic data, a second group, for example, transcriptomic data, and the different different groups. And and some the patients with different ages. We learn aging process, and we think what the aging process is similar in one group and another group because it's Homo sapiens in one in first groups, and is if how you know for what your opinion about using the similar approaches for different types of different groups of patients.
Well, so if we have different groups of patients, if we have like for all of them similar multi-omics data, therefore there is no problem to incorporate. But if, as you said, you have different omics data for each group, it's better to do like meta-learning or transfer learning using some previous data, maybe in open databases where the similar multi-omics analysis was done for also some groups of people. Of course, it's not too good, I think, in one model incorporate different multi-omics data for different groups. So you can have different groups, but similar multi-omics data for them, for example, but not like very different multi-omics data, different for each group.
I think it's if exists some latent space in which some which the process of aging may act in the latent space, and this latent space, the some types of the data, some types of omics data can transform to the similar latent space. And it's not the reason to use some type of data for some different groups or not.
Well, of course you can transform the omics data to latent space, but still, if you, for example, transform transcriptomics data to latent space, this latent space will still talk you about the transcriptomics. And when you, for example, for other patients transform metabolomics to latent space, it will still be low dimensional as for the first one, but it will talk you about metabolomics. So for if you want to like integrate the first group of patients and the second, but for first you measured only transcriptomics and for second only metabolomics, you should I think choose some complicated things like transfer learning or meta learning, but not just direct integration.
Okay, thanks.
Colleagues, do you have any other questions? Because I have a lot. Yes? Okay, let me ask a question about meta-learning. I really wondered about deep omics approach. Did they use data from different sources for integration multi-omics data? And this is the first part of the question. The second part, do these sources contain data of multiple modalities simultaneously? Or we can integrate data within one neural network, just taking data from different sources and doing partial training steps, having data from different sources like this.
So in this meta-learning, okay, in maybe in some meta-learning articles they did some other things, but with which I am acquainted, they, for example, took different data sets, as you told, but they took it from like one source, from pan-cancer, TCGA pan-cancer, and here for all these data sets, the similar multi-omics analysis was done. So for each, for example, it can be cancer, breast cancer and lung cancer, but still for this cancer and the patients with this cancer and the patients who are those cancer, you did all the omics part, both transcriptomics, both proteomics, and both clinical data. So this data, in fact, it is similar. The only difference is the type of cancer. Well,
Yes, that's an answer. As I understood, they use the database for cancer, and this database contains a lot of multi-omics assays simultaneously that can be used for training the same neural network. That's the opposite situation to what we can see in aging domain, where each aging experiment is typically associated with one omics data, like transcriptomics of aging, metabolomics of aging. And as I know, there is no databases containing multi-omics data related to aging. Am I right?
In GeoDatabase, there are some datasets which have metadata. For example,
Aging multi-omics datasets.
No agent. Some samples for transcriptomics, for example, and each samples has also the metadata with some blood parameters. It's a type of
yeah.
example.
Yeah, that's a good point, actually. Yeah, I didn't I didn't find any data set with simultaneous transcriptomics and blood parameters of different ages. That would be cool to see this one. If you can, Alexei, could you send it me? Because
Okay.
I really interested in such data set. It's because it's really part of multi-omics story to compare transcriptomics and clinical parameters. And okay, my question was
may I just input? I think that there are really not so many databases where there's simultaneous multi-omics data, and as for example in TCGA there is multi-omics data in the majority of articles. You can see this database. So like the data from this database, it's analyzed hundreds of times with different methods, just because it is one of the few databases which includes all this data simultaneously.
But as I understand, there are no data suitable for survival analysis of aging. I mean, the survival times versus multi-omics data. Like this, such data is something outstanding. We can't see this currently.
Survival times, you mean, and the multi-omics data?
Yes, survival times, but for all cause mortality, not for a specific cancer mortality and for all cause mortality. Do we have such population survival data data sets combining multi-omics data and survival times from all cause mortality?
So again, here we can have clinical data, and in clinical data you have survival data and survival times. So the time, or maybe I just didn't get the point.
Yes, but are these patients control patients or cancer?
No, only cancer patients.
Yes. I wondered if there's some data for control patients.
No, unfortunately, no. I think that the course is just commercially problems.
Yeah. Okay, and I have a small question related to slide 13. Yes, I'm interested. What is Bayesian supported PCA? It is like just a probabilistic PCA. I didn't see such name. I faced with probabilistic PCA method, but what is MENSI? What is Bayesian supported PCA?
Just a second. In fact, about this method, I learned from from the part of the article where they told about the similar researches, and I didn't acknowledge with it a lot, so I didn't too deep in it, because it's like statistical approach, and I mostly wanted to focus on deep learning approaches. Yeah, but in fact, yeah, it's something about the probabilities, and